Hi, everybody. Welcome to Microbes in the Modern World, From the Globe to the Gut. This has been a wonderful seminar series this term, and this is the last talk in this series. So before we get started, really, I'd like to take just a moment to thank our sponsors. The University of Oregon's College of Arts and Sciences has chipped in a lot to help us put this on, as well as the associated students of the University of Oregon. So that's all of you, or most of you, a bunch of you. The Department of Biology, the Institute of Molecular Biology, the Institute of Ecology and Evolution have all chipped in, and the Oregon Humanities Center's Endowment for Public Outreach in the Arts, Sciences, and Humanities has chipped in. Right? Tons of people. So let's thank all of these people for helping to put this together. And GREEBS. This is the Graduate Ecology and Evolutionary... Why can't I say that? The Graduate Evolutionary Biology and Ecology Students. That's our student group. We as budding academics feel very strongly that it's important to get science not just here in the ivory tower, but out to the people. So this is why we sacrificed our spare time and our sleep to put this together for you. So this couldn't have been possible without the combined effort of all of the graduate students in this department. So, microbes in the modern world. So this seminar series was designed to answer some basic questions about microbiology and to bring to the public some of the understanding that new and emerging technologies have given us about microbiology. We wanted to answer some basic questions like what are microbes and what do they do and where do they live? And address issues ranging in scope from the environment and global ecosystems all the way down to your own guts. Luckily, right here at the University of Oregon, we have some of the top minds working in this field. So we're able to bring you not just science, but science that's being done right here on our very doorsteps. So, on the 17th, Dr. Bohannon gave a wonderful talk, a scientist in Whoville, New Perspectives on the Invisible World of Microbes, and he told us something about how microbes aren't necessarily germs, how they're not necessarily bad, and how they're parts of ecosystems. And he's used his own work to illustrate how when we change ecosystems, we change the microbes, and how changing those microbes can have really drastic effects. And then on the 8th, Jessica Green came and gave us a talk on the ecology of indoor environments, how buildings are necessarily habitats not just for us, but for the microbes we bring with us. And her work aims at figuring out what lives in buildings and what can we do to buildings? How can we build in a manner that might encourage the growth of microbes we like and not microbes we don't? And I will remind you, both of these talks, even though they've already happened, if you missed them, you can still watch them. They're available online as webcasts at this web address. And Karen's talk after today will also be online. So if you want to remember something and you can't quite remember it, you can always watch it again. Which brings us to today's talk. Dr. Karen Gilliman will be delivering molecular dialogues with the microbes inside us. And she'll be telling us something about the microbes that live in our guts and how we figure out what's going on in that relationship. So let me tell you a little bit about Karen. Dr. Gilliman was an undergraduate at Harvard and Radcliffe colleges, where she got interested in doing research, and then she was interested in development, so she did her PhD at Stanford University, studying organ development using fruit fly model system. And then that kind of got her interested in how the environment affects development, particularly how microorganisms can play a role. So she went on to do a postdoc, also at Stanford, studying the interaction between bacteria and hosts. And then in 2001, she moved to our wonderful institution here and decided to put together development and host microbe interactions. And she developed this amazing model system, this germ-free or microbe-free zebrafish model that she can use to study how microbes play a role in development. And I'll let her tell you a little bit more about how she's doing work with that and what she's doing. And before I turn it over, Dr. Gilliman is an extremely distinguished professor. There's a couple of awards I'd like to highlight. I can't give the whole list, it's a little too long, but there's a few things. that I think are worth mentioning. She recently gave the annual Ari Dyer Lecture for the NIH, the National Institute of Health. And this is a lecture that's been going on since the 1950s in honor of Dr. Rolla Dyer, who's an authority on infectious disease, and also head of the NIH in his day. She also recently received the NIH's National Institute of Diabetes and Digestive and Kidney Disease Institute's 60th Anniversary Early Career Investigator Scholar Award. So these are very prominent awards. Dr. Gilliman is wonderful, and I will turn it over to her now.
Okay. So can everyone hear me? Great, thanks. So first of all, I want to thank Rue for that very nice introduction and Greaves for organizing this really exciting seminar series. And it's been a real pleasure to attend the other lectures, and it's a pleasure to be here today. And today, I have a number of things that I'd like to accomplish in our time together. I'd like to tell you a little bit about the history of how we've studied how humans are ecosystems for microbes. So in this series, we've been thinking about microbial communities globally on our planet, within the places where we live and work. And now we're going to be talking about the microbial ecosystems on our bodies. And we're going to start by thinking about how this has been a discipline in the history of science. And then I'm going to be telling you about how a very exciting revolution in the technical advances that have allowed us to describe microbial communities has really changed the way we're thinking about the microbes that we're associated with. So to give you a little bit of the current understanding of how humans are associated with microbes, but really that this is a very nascent body of knowledge that raises many pressing questions, and I want to introduce you to some of those questions today in the field of human-microbe interactions, and show you, what are the burning questions that we're really excited to answer in this field. And then finally, I'm going to give you just a taste of some of the experimental approaches that my own lab is using to answer some of these questions. And we're doing this in a model organism where we can perform experiments that would not be possible in humans. So I wanted to give you also a flavor of what we're doing. So when we think about our, the current knowledge of who we are as humans today, we're at a very exciting time in human history. And the next image, I think, is large, so it's taking a little while to advance. But I want to just take a moment to reflect on the fact that we have an enormous wealth of knowledge about ourselves that was not available just a decade ago, and that's the knowledge of our entire genetic makeup, and it's becoming possible with advances in DNA sequencing technology to really understand the genetic makeup of ourselves in ways that was not possible just recently. So I think I'm going to have to escape here, sorry, and just get to the next slide. Okay. So when we think about ourselves as humans, we have this new understanding of who we are based on all of the genetic information that's encoded in our DNA. So every single one of us is made up of a whole collection of 10 trillion different cells. Each one of those cells contains a blueprint of how this organism is going to function, and that consists of about 25,000 genes. And so it's now, with advances in DNA sequencing technology, getting to the point that very soon everyone will know their entire genome sequence. This is becoming technically possible. And yet that information does not encompass all of the genetic information that dictates our biology. If we consider the cells that make up this organ, pictured here in this drawing by Leonardo da Vinci of the human digestive tract, there are in fact 10 times as many bacterial cells resident within this organ than all of the cells that make up our bodies. And this is a very diverse collection of cells consisting of approximately 10,000 different bacterial, 100,000 different bacterial species, and 1,000 bacterial species, but then also many eukaryotic, archaeal, viral species as well. So it's a complex ecosystem. And if you think about the collective information encoded within that, the collective genomes of all those cells, it really dwarfs the size of the human genome. So even though we're making rapid advances in DNA sequencing technology, it's still sort of a farther frontier that will in the future be able to know all of the DNA that's encoded in all of the cells within us, because that includes this complex microbial community of cells. It's perhaps not surprising if we think that we're habitats for microbes. When you think about the history of life on Earth, that for the last 4 billion years, the Earth has been inhabited by microorganisms, and it's only really the animals have been recent newcomers to this environment, and so animals have evolved within a microbial world, and so then it makes sense that animals have evolved to have strategies to coexist with microbes. But it's in very recent history of humans that we actually have become aware of the existence of these microorganisms, and that required technical advances in the ability to magnify cells to the point where we could see microscopic organisms. So it was the Dutch merchant Antonie van Leeuwenhoek, who developed methods to create lenses that will allow him to create this very simple handheld microscope that you see here, where the specimen is simply just placed on a screw that's screwed over a lens, as shown here. And it was with this new advance in optics that he was able to visualize for the first time single-celled organisms, microscopic organisms. And it's interesting that very early on in his discovery of these cells and his investigations of these cells, he immediately started to ask, are these types of cells associated with me? And he was interested in host-microbe interactions, and he looked and examined whether these types of microorganisms were present in his saliva, on his skin, and in his tears. And so there was this fascination about this microscopic life form and its association with humans from the very beginning of the history of knowing about the existence of these cells. And when you think about it, this was really a radical finding when he was able to observe these types of microscopic cells on himself. And it was sort of a change of thinking about the human body as a rather serene landscape to one that's just incredibly densely populated with life. And that's an idea that took a while for people to recognize, and it's still something that I think each of us today is kind of a miraculous thing to realize that we're actually habitats for so many microbial cells, that we're just teeming with life. And then there are all sorts of questions about the consequence of this association. And of course, at this time in the history of Europe, there was a very intimate familiarity with infectious diseases, but not yet known to be infectious diseases. So this is another painting by Broyles that shows the devastation of the bubonic plague throughout Europe that decimated the population of Europe and was still very much in people's minds several centuries later. But the connection between microorganisms and possible disease was actually one that took several centuries to develop, and it was really not until the 19th century that the pioneer microbiologists Louis Pasteur and Robert Koch coined this concept of the germ theory of disease and proposed the idea that these microorganisms that people had come to be able to visualize and even to be able to culture could in fact be causes of diseases, and that could explain the infectious nature of certain diseases, that they could spread because they were caused by agents that could actually proliferate and grow. And these and disciples of Pasteur and Koch developed methods to identify culprits for many different kinds of diseases. And these are actually original micrographs taken by Robert Koch, showing a pure culture of Bacillus anthracis, the agent of anthrax. This was part of his demonstration that indeed this particular microorganism could cause this disease in experimental animals. But even though people had come to realize that microbes could be agents of disease, they had very little, there still were few tools available to manipulate these associations. And so this is a drawing, a painting of By the Deathbed by Munch, fever, showing that infectious diseases causing diseases such as fever and death were still very much of a concern among the population. Even though they realized that these could be caused by microorganisms, there wasn't necessarily methods to try to combat these types of diseases. And that required additional advances with the discovery of antibiotics by Alexander Fleming, who discovered penicillin in 1928. There was an enormous excitement about the ability to actually combat these organisms that could actually cause diseases, as shown in this advertisement from in World War II. And so there was, I think, when people thought about antibiotics as a new tool for medicine, they could imagine, how this was something that could specifically slay a particular foe, as shown, as demonstrated by this depicted in this image of a bullfight, where you could have this targeted therapy that could eliminate a culprit for a disease. But one thing that I want you to be thinking about in this lecture today is that when we think about the kinds of treatments that could target a particular microorganism, there's going to be collateral damage associated with that. And so as we start to consider our associated microbes in the context of an ecosystem, when you think about the consequences of treating, targeting a particular member of that ecosystem, there's going to be additional ravages of war associated with that. So in this series, we started out this series with a wonderful talk by Dr. Brendan Bohannon, where he introduced the exciting times that we're in right now that are allowing us to describe the diversity of microorganisms in our environments in ways that have previously not been possible. So we have now molecular methods that allow us to determine the DNA sequence of biomarkers within communities of microorganisms and survey who's there without having to cultivate those organisms in the laboratory. And that's given us an enormous new sense of the kinds of diversity of microorganisms in our world. And he shared with us some of the work that he's been doing looking at microbial communities in soil samples and shared with you the abundance of different kinds of bacteria that are present even within a single gram of soil as revealed by these new molecular techniques. And he raised the important question about whether human activities that we know are altering the environment at macroscopic scales are also potentially altering microbial communities and perhaps leading to extinction of microbial species that we're just very recently becoming to get to know and appreciate through these new molecular methods. In the next talk in this series, Dr. Jessica Green told us about the ecology of indoor environments and work through the Center for Biology in the Built Environment that she directs. And she showed us how studies of the microbial communities associated with environments such as a hospital are revealing a diversity of different microorganisms that are there and starting to raise questions about how we design our built environments and raising the question of whether we should be rethinking the design of our the buildings we inhabit to better foster healthful microbial ecosystems and to try to promote health and well-being. So what I wanted to share you about with you today is a growing excitement about looking into the ecosystems of microbes associated with humans. And part of this effort is driven by an effort funded through the National Institutes of Health called the Human Microbiome Project, or HMP. And the goal of this project is to describe the microbial ecosystems of humans, looking at the diverse microbial communities that are present throughout our bodies, on all of our mucosal surfaces, on our skin, and inside our digestive tracts. And we're starting to get a picture of the kinds of microbes that are present, as illustrated in this review article by my colleague Ruth Ley at Cornell, where she's depicted some of the findings from this Human Microbiome Project, which is really a survey of the diversity of microorganisms throughout the body of various human subjects. And what are shown here are pie charts representing some of the major bacterial phyla or groups that are found at some of these different habitats on a human. And you can see that in different places throughout the body, you find different consortia of microbes. And one thing I should emphasize, that this is a summary of many results of surveys of many people, and one of the major findings of the Human Microbiome Project has been the individuality of everyone's microbial ecosystem. So this is average across multiple individuals, but if you were to look at any individual and compare that person to another individual, you'd see that there's quite a lot of variation between individuals, and that in many ways our associated microbial communities are as distinctive as a fingerprint or as our own genomes. So we're starting to get this picture of what kinds of microbes one might find at any moment in time associated with a human, but really this is raising many more questions than it answers. Immediately when we see this depiction of certain microbes associated with us, we wonder, well, how did they get there? What factors determine the assembly of these microbial communities? And one could postulate, for example, that the exposures one has could influence this microbial community, that one's diet could potentially influence the microbial communities resident in the digestive tract, which is the place where all of that ingested food is input. And then also the genetic makeup of a particular host could determine the microbial community composition. And these are still very open questions. I'm going to give you a little bit of a flavor of some of the ways we're trying to answer these questions, but I want to emphasize that we're just at the cusp of understanding how to address some of these questions. So there's still there's a great deal that we don't know. We're also, another question that's very pressing and interesting is to ask how stable these gut microbial communities are over time, both in terms of any particular person's lifetime. Are they fluctuating in a matter of minutes, hours, weeks, years through one's lifetime? And also across generations. Are the microbes that we're associated with today the same as the microbes that our parents or grandparents were associated with, or that early humans were associated with? And these, again, are questions that we are just beginning to get a glimpse of the answers to. And then finally, a really interesting question. We now know something about these resident microbes, but we're wondering, does it matter in terms of our health who these residents are, and do the changes in these microbial communities, do they actually have the potential to drive diseases? So we've seen that in the history of studying host-microbe interactions in humans, we have understood the concept of a single pathogenic microorganism causing an infectious disease. But now should we be thinking about how overall changes in microbial communities might actually drive diseases, and how can we address these questions? So I want to give you just a little bit of a flavor of some of the answers that we're getting to some of these questions from looking at humans. And one has to do with how we acquire our resident microbes. And there's a lot of interest in this. And this is just one piece of data that suggests that the birth event is actually a source of inoculation for a newborn infant. So the fetus is sterile, and the first acquisition of associated microbes comes at birth. And this is a study that looks at samples of microbial communities from many different individuals. And what's plotted here is a distribution of those communities relative to their variation, so that communities that are more similar to each other in their composition are clustered together on this two-dimensional graph. And what you can see is that there are a number of samples that have been taken from mothers who were entered in the study, including in red, they're vaginal samples, and in blue are skin samples. And then there's subsequent to the mothers delivering babies, the newborn babies were also sampled. And what's shown here is a distinction between babies who are delivered vaginally versus by cesarean section. And what you can see clearly is that the babies born by a vaginal route have more similar communities to the mother's vaginas, and the babies that were delivered by cesarean route have a more similar community to the skin. And actually what's interesting is that the babies born by cesarean section are no more similar to their mother's skin microbial communities than the skin microbial communities of other people who are in the room, the nurses and the doctors in the operating room. So this tells us that early inoculations can influence the microbial communities in a newborn. But how do these communities then develop over time? And one thing that's actually, I think, a very exciting finding is new evidence looking at the composition of breast milk, which is a natural food for humans, and finding that it actually contains sugars that are not digestible by human cells, but are in fact an excellent source of nutrition for gut microbes. So mother's milk actually contains food to nurture a gut microbial community in newborns. This is a study of just a single infant over time, and it's looking at at various days after birth, looking at the composition of the microbial community and correlating that with different events in this infant's life. So this is just a natural history of a single human, but it shows you some trends, that there's changes in this community over time, and one sees that there are in particular changes in the introduction of this group of bacteria, the Bacteroidetes, that occurs after the introduction of table food and formula. So this also lends some credence to the idea that a diet does influence the microbial community, at least in this one particular individual. The correlation between the composition of one's diet and the composition of the microbes is one that's very fascinating, and it's a difficult one to study in humans because humans have such varied diets, and it's actually quite difficult to get human subjects to subsist on really defined diets. So there are lots of studies that have been done in volunteers, and then a lot of times people sort of suspect whether the subjects were actually cheating and not eating what they were told to eat. So it's a lot easier to do those kinds of experiments in experimental animals where you can control what they're ingesting. And this is just one kind of experiment that starts to get at this relationship between diet and microbial community composition. And this was one in which laboratory mice were raised on either a normal mouse chow, abbreviated CHO, which is basically a plant polysaccharide-rich diet, versus a diet, a Western diet, which is high in fats and simple sugars. And what you can see is that in these groups of experimental mice, at least, there are changes in the microbial community that are correlated with these different diets. In particular, if one looks at the representation of two different phyla, the firmicutes and the Bacteroidetes, you can see that there's a real loss of the Bacteroidetes and a depletion of that group in mice fed on a Western diet. And another important parameter of these microbial communities is not just membership, but complexity of the community. How much diversity is present? How many different types of organisms are present? And one measure of that is shown in this panel B here, where it's using what's referred to as a Shannon index, which is one measure of diversity. And there's various ways that one could look at this, but overall, what I want you to take away from this is that the mice that were ingesting this plant polysaccharide-rich diet, the CHO diet, have a much higher diversity than the mice that were subsisting on a Western diet. And so that's another important parameter to consider when we're starting to think about microbial ecosystems. In general, one of the principles we've learned from ecology is that more diverse communities are more resilient to changes, environmental changes, and so one would tend to think of a more diverse community as a more healthy community. So have changes in our human ecology been changing our associated microbial communities? So I've just showed you some data that subsisting on a diet that's very high in fats and simple sugars can alter your associated microbial communities. And there are many ways in which our associations with microbes have been changing in our very recent history. We have a much more sterile environment now, and there have been very important advances in public health that have arisen from the realization that microbes can cause infectious diseases, and so that's changed the way that we practice medicine, and those have been important advances. But it has changed the kinds of daily interactions that we have with microbes. We live in a much more sterile environment. The kinds of foods we eat are much more sterile and much more processed. And so that's another sort of daily change in our associations with microbes. And as I mentioned also, changes in birth and birth route. So now a third of U.S. births are by cesarean section today, and that's a change from 100 years ago. Changes in frequency of breastfeeding. All of these kinds of things can change how human populations, especially in modernized society, are associated with microorganisms. And we don't fully know the consequence of this. We don't have a really good historical record of what the microbial communities were in people 100 years ago or 10,000 years ago. But there's increasing interest in the possibility that changes in our associations with microbes are changing the kinds of microbes we're associated with in ways that could have effects on our health. And one formulation of that idea was put forth by David Strachan in 1989, which was referred to as the hygiene hypothesis. And this is something that I think is now sort of an idea that's quite common in the public, which is the idea that if we live in a much cleaner environment, maybe we have fewer encounters with microbes that would normally stimulate our immune systems and promote healthy development of our immune systems. And so in the absence of these stimulations, then we're at risk for developing different types of diseases, potentially autoimmune diseases or allergies or other things where there seems to be an overactivation of the immune system to innocuous stimuli. And this hygiene hypothesis has been definitely something that sparked a lot of interest, and people have been working on really kind of clarifying and formulating those ideas. And I think an even more exciting and compelling version of this idea is a variant referred to as the disappearing microbiota hypothesis, where microbiota refers to the microbial community associated with any habitat, but in particular here, the habitat of humans. And this posits that there are changes with our associated microorganisms that could be leading to increased incidence of certain diseases. But it's more than just the random interactions with microbes in the environment. The idea is rather that we're actually losing important resident associates that historically have been inhabitants of humans and are now being lost from our environment. So it's more of an idea of extinction of particular microorganisms that have been important in the past in our normal development and health, and we're now losing those through changes in the way we interact with microorganisms. So this harkens back to some of the ideas that Dr. Bohannon raised about the possibility of extinction of microbes in our environments. Maybe we're particular human-associated microbes are becoming extinct, and this is could be have serious health consequences. So I've raised the possibility that changes in the microbial communities could be associated with diseases. Well, what kind of evidence is there that an altered microbial community could be actually causing disease? And some enticing evidence is emerging from these same sorts of studies of the Human Microbiome Project and other types of studies, where we're now being able to survey the microbial communities, both of healthy individuals and of disease patients. And again, there's an enormous amount of heterogeneity in the kind of data that one observes when surveying microbial communities associated with different individuals. But there's starting to become a picture emerging where there are characteristic shifts in the composition of microbial communities associated with patients with certain types of diseases. So, for example, patients who suffer from inflammatory bowel disease, excessive inflammation of the colon and the intestine, are characterized by having altered microbial communities with an increased prevalence, for example, of these proteobacteria shown in the blue part of that pie chart, and a relative loss of the Bacteroidetes groups. There are also similar shifts that one sees with type 2 diabetes, and then disease of early infants, and especially premature infants, necrotizing enterocolitis, is also associated with a real expansion of the proteobacteria. So these are intriguing correlations when seeing some consistency and shifts in the microbial communities found in particular patients. And some of these are diseases where the microbes are, in close association with the affected tissue. For example, all of these have the digestive tract is an important organ affected in these diseases. But really, these are just correlations. And how do we get at causation? How could we actually demonstrate that an alteration in the microbial community could really drive disease progression? And that sort of demonstration would be required to take the next step of trying to think about how to treat these diseases as diseases of altered microbial communities. So what I want to now turn to is telling you a little bit about work in my own research group that tries to address some of these questions using model systems. And so we can use laboratory-reared animals where we can manipulate the associated microbes and start to actually get at looking at issues of causation and not just correlation between these microbial communities and the host traits. And what I'm going to be telling you about today is, in particular, the zebrafish as a model system that we use. And we're particularly excited about the zebrafish because it's a system where the zebrafish has many attributes of humans in the way that their digestive tracts and their immune systems develop and function. But it's very amenable to manipulations, and in particular, we can raise sterile zebrafish. So what I'm showing you on the bottom are micrographs where I'm showing a slice through a zebrafish digestive tract, and in red are illuminated the bacterial cells in that preparation. And you can see that we can make sterile, or what we refer to historically as referred to as germ-free animals that are devoid of all bacteria. And then we can also take those germ-free animals and re-inoculate them with either complex communities of microbes or individual bacteria and bacterial strains, and we can look at the consequence of those associations. And another real advantage of the zebrafish is that they're transparent, and we can visualize host-microbe interactions in real time. So what I'm showing you here is a movie of the digestive tract of a zebrafish larva. In red are individual bacterial cells in the digestive tract, and in green is a zebrafish immune cell that's kind of surveying the landscape here and watching as these different microbes, these bacteria, you can see those bacteria sort of taunting the immune cell there. In this. So we can really study these kinds of processes in live animals. And what I want to tell you about today is just a short bit of a current investigation where we're looking at what happens to gut microbial communities when you change aspects of the habitat of the digestive tract. So what I just showed you is a movie, I'll show it again, of the digestive tract of a zebrafish, and you can see a peristaltic wave that is going through. You can also see blood cells are streaming above there. And then in the stained micrograph above, I'm showing you the cells that are responsible for that gut motility, that peristaltic wave of movement. And those are the neurons that innervate the gut and regulate the contractions of this organ. And in collaboration with my colleague Judith Eisen here, we've been studying a zebrafish mutant that lacks those neurons that normally innervate the gut. And what one finds is that the intestine is unable to have these peristaltic waves that go through. So it's a very different ecosystem which lacks the normal motility and flow through that system. And another demonstration of the dysfunction of this organ is shown here, where we have fed our larval animals fluorescent foods. And we can do this where we first offer them a green fluorescent food, and then for a short period of time a red fluorescent food. And you can see with the normal, or what we'll refer to here as wild-type animal, it's able to efficiently clear the green food. And so now, viewing just a few hours after offering the red food, we see a red gut that contains red food. Whereas this mutant that is unable to carry out these peristaltic waves along the length of the gut has a bolus of green food still stuck in the gut. So you can imagine that this is a very different ecosystem in this mutant versus this wild type. And one thing that we looked at is whether this was an ecosystem that was more diseased, and whether there was inflammation in these guts. And a way that we can measure that is we count the number of immune cells that are come into this organ. And that's easy to visualize with this particular stain, where you're looking at cells, brown cells. Actually, do I have a pointer that I could? Okay, but the brown cells on the length of the gut, and we can also quantify this. So I'm showing you just our quantification, where we're counting the number of these immune cells in a gut, and what we find is that there's an increased number of these cells in the mutant versus the wild-type animals. And so we wondered, well, is this inflammation that we're seeing in these mutant zebrafish due to the microbes that are in the gut, and is it an inflammatory reaction to those microbes? And one way that we can do this is we can take our animals that we know would normally have this inflammation and derive them under these germ-free or sterile conditions. So if the inflammation is just inherent to the mutant, the nature of the mutant, then we would see that even in the absence of gut microbes, we should still expect to see inflammation as depicted in the schematic on the lower option. But on the other hand, if it's the microbes present are driving the inflammation, then we should be able to alleviate that inflammation if we rear these animals under germ-free conditions. And that's what we found as shown here, where what's important to just look at is that the mutant under germ-free or GF conditions has the same number of these immune cells in the guts as the wild-type animals under the germ-free conditions. So this is an inflammatory response to the gut bacteria. And then we wondered, well, is this community of gut bacteria different in the mutants versus in the wild-type animals? Are they actually experiencing an altered community? And one way we can do that, look at that, is using the kind of biomarker DNA profiling experiments, and we do see differences doing that. But we also wanted to sort of ask this at a much more functional level. And we can do this ask this kind of question by doing what we refer to as a microbiota transplantation experiment. And what we do is we take the gut microbial community from an inflamed mutant, where we know that we see the inflammation in the mutant. We can harvest those microbes, and then we can introduce that into a wild-type animal that's sterile, and allow those gut microbes from the inflamed gut to colonize the sterile wild-type animal. So we know that normally the wild-type animals don't develop inflammation. And so then if the gut microbes from the inflamed mutant are really not any different from what we would find in a wild-type animal, we wouldn't expect to see any inflammation when they're colonizing a wild-type animal. However, if this gut microbial community is really different, it's altered in its properties in some way, we should see then that it's able to induce inflammation in even a wild-type recipient. And that's the finding that we found from these experiments. When we perform this experiment where we have a germ-free wild-type recipient, and now it's receiving the microbiota from a wild-type animal, it has less immune cells present in its gut than if it receives microbiota from this inflamed mutant donor. So that's telling us that the microbial community itself is driving the disease. So this is similar to an experiment where you would inoculate with a single pathogen and see an infectious disease. Now we're seeing this is a transmissible infectious disease, but it's in a whole community of microbes that together collectively are driving the disease. So we can think about this as that the gut environment here is influencing the microbial community that assembles there, and that in turn can influence gut health, the state of inflammation, which is intimately then fed back into the state of the gut environment. And so you can see how there's these reciprocal interactions between the host and the microbial community that influence the composition of the community and the state of health of the host. So that gives you a little bit of a sense of the kinds of experiments that we're doing in our lab now to address, what are the microbial communities associated with animals like ourselves, what are their functions. And so I want to make sure to leave time for questions, but I thought I would just end, what does this mean for any of you? What does it mean for me? And I want to emphasize that we really are at the very beginning of our knowledge of what these community compositions are, how they assemble, what they're doing. And so, if you wanted to take knowledge from what I've told you today and apply it to your daily life, I'd say that there's still many things that we don't know about. For example, the efficacy of probiotics, prebiotic diets. There's a lot of promise in these areas, but I think there's still much work to be done to really validate whether particular, for example, probiotics are efficacious for treating certain diseases. But I think one can start to just think of, it's helpful to think about the microbes associated with you as an ecosystem. And so there are just some simple things that you could do to try to nurture your gut microbes, and sort of, just common sense things. Just one responsible thing would be to use antibiotics judiciously, to realize that you're treating an ecosystem, not just a single particular pathogen when you use an antibiotic. And so it's something to give thought to. There's a lot of questions about what kind of diet would be healthy to eat. And Michael Palin, of course, has said that you should only eat a diet that your grandmother would recognize as food. And I think it might be a good idea to try to eat a diet that your grandmother's microbes would recognize as food. So, I think that's a good way to nurture your microbes. We can think about, nurturing our environmental microbes as those important sources of inocula for our own host-associated microbes. And so, again, these are just sort of some simple, suggestions that it's important to support the responsible use of antibiotics in agriculture and animal husbandry, and just to think more carefully about how human impacts on our environment are impacting our microbial environments, and the possibility that we might be losing important microbes to extinction before we even really get to know them. But finally, I would just like to encourage all the young people in this room who are thinking about exciting careers, that I think that microbiology is an incredibly exciting future career, and also, but for all of us, we all are ecosystems for microbes, so it's worthwhile to get to know them and to know more about them and to continue to follow advances in research about microbes. So I'd be happy to take questions. Thank you. Yeah, so the question is whether there's experimental support for the hygiene hypothesis. And there's definitely an enormous amount of support for the idea that in the absence of any associations with microbes, you have a different immune system. So people have been looking in germ-free animal models at immune system development, and it's clear that it does not occur normally. And some of those shifts, some of the are consistent with the idea that with models that those germ-free individuals are now hypersensitive to developing certain kinds of diseases like allergy or autoimmune diseases. So they lack certain immune cells that normally keep some of the overactivation of the immune system in check, and those are absent. So there's a lot of support for that idea. But those are, that's an extreme situation where you have a completely germ-free experimental animal. So then taking that knowledge and trying to apply it to humans is a little bit more complicated. So, where humans are not germ-free, but it certainly makes sense that we're reducing our associations with microbes. But this refinement of the hygiene hypothesis, the disappearing microbiota hypothesis, is I think one that's a little bit more thoughtful and compelling, because it says it's not just any random associations with microbes that we might be reducing by using hand sanitizers, but perhaps that they're key microbes that we're having fewer interactions with. And so that's something where one could imagine more thoughtful types of therapies to promote normal development of the immune system. If there are certain particular members that we want to make sure are residents within us, and that idea of a probiotic where you could actually try to promote associations with particular microorganisms is one that's quite compelling. Yeah.
Karen, I was wondering, have there been any studies, or have you heard about anything about archaeological finds from like people or ice? Have they tested their guts?
Yes, there have been some studies where people have tried to profile the bacterial resident members of feces from or gut contents from mummies and also frozen ancient humans. And as you can imagine, these are enormously challenging things to do, because the risk of contaminating with your own skin bacteria is enormous, and it's hard to demonstrate for certain what's who's there. But so given that these are not terribly great quality data sets, it still looks like they're pretty similar communities that are associated with ancient humans to modern humans. But when I say pretty similar, it's this looking with very hazy lenses. So it's not going to be able to detect the kind of variation that I was showing you that one might see between a healthy subject and a diabetic subject or something at that level of resolution. So, but it doesn't look as if we have entirely different microbial communities. But those are hard studies to do.
So in that vaginal versus cesarean delivery, what's the time course for that sampling? And do the kids start to become more like their mom over time, or should people
Yeah.
start getting inoculated with vaginal swabs or something?
Yeah, so that was 1 week after birth. And at that point, they actually were just sampling the skin of the newborns, and that's not really distinguishable from the gut contents. And they didn't, in that study, follow the kids over time. There have been a number of studies where people have tried to do, sort of long-term looking at, for example, risk for inflammatory bowel disease in, adults who were either delivered by cesarean or vaginal birth. And there isn't a strong correlation. There are some examples if you look at the data in a certain way, and it's a little bit stronger correlation with males versus females, that there's a slightly higher risk for IBD and cesarean delivered, individuals. But it's those, clearly those communities do converge, back, and other influences influence that assembly. But I think it's still really very much of an open question. Yeah.
That one chart you had where you graphed out the infant, you had the introduction of formula, and then just about 2 clicks down there was the introduction of amoxicillin. Now, on the formula is the reason why, the yellow spiked up, the same reason why the amoxicillin went up, the antibiotics in the formula was killing the good gut bacteria?
That's a great question, and the thing is that was just one infant. So, we can't know. This is all just, yeah. So there have been some studies that have looked at what happens if you just dose, healthy volunteers with antibiotics. And what one sees is that, by and large, the communities shift back to the gut microbiota communities shift back to what they were prior to the dose. But there's a little bit of a change. And what's interesting is that's resilience or ability to return back to the normal previous state varies across individuals. And then they also, in a further follow-up study, did a double dosing experiment where they dosed once and then waited 6 months and dosed again, which is not really, a medical doctor would not think that that's such a radical treatment. But what was observed in that study is at post-second dosing, the communities were really much more perturbed and didn't restore back to normal. So that would suggest that perhaps after the first dosing, you've lost a little bit of the resilience and the ability to go back to normal, and then further perturbations can further exacerbate that. But these kinds of studies are difficult because every individual is going to have a different diet, genetic makeup, life history, and so there's a lot of variation across individuals in the studies. Yeah.
When it comes to restoring balance that you need, so that you have an antibiotic that would target a specific one, based on what you were saying before, then it would be difficult to restore it because it wouldn't just come snapping back, right? Is that what you're saying?
I was suggesting that there can. So in general, in the study, that one found that there was quite a bit of restoration back to the normal community, but it wasn't perfect. And so then when you think about the average number of antibiotic courses that typically people have, you can imagine that you could be eroding some of the resilience of that community over time. Yeah. So there are certainly questions about how do you what should you do after you take antibiotics, and I think it's not a bad idea to take some over-the-counter probiotics, and that those are probiotics that are currently available for purchase today, have the status of being health supplements. They're not medicine. So they've been validated that they don't cause terrible risk for ingesting them, but they don't actually, there's no requirement that they actually cause any health benefit. But it's, anecdotally, many people find that probiotics can be helpful, but there's sort of, I think we're just really at the infancy of understanding what are useful microbes to ingest as try to promote a healthy gut community. Yeah.
Have you done any studies where you look at some of the same time frame, like some of the antibiotics, and you look at them, some of you not have antibiotics, you look 6 months later and see how much change there is, and also with some of you doing one of the
Yes. When you look at people, in general, if you look at people who haven't had any kind of changes with antibiotic treatment or diet, the microbial communities are really quite stable over time. There's some evidence that those communities become less stable in old age, and they're also less stable in early life. And so that sort of also correlates with changes in immune system function and other things. There definitely are examples where of following people who've gone onto altered diets and seen very quick, rapid shifts in the microbial community. But one of the things I should emphasize is that if you could see changes in the membership of the community, but that still doesn't necessarily say that the community is different in the functions it's carrying out. And so that's a whole new, additional complexity that we're just, starting to learn how to address. Now that we're starting to know who the microbes are, what are they doing? That's a really big question.
Is there any evidence that the actinobacteria inside us have any antibiotic function like Streptomyces?
There's actually an enormous excitement about mining the gut microbiome for novel antibiotics. I don't know of any particular example that's been really clearly demonstrated, but when one looks at the genome sequences, there's sort of a lot of capacity of genes that could encode novel antibiotics. So there's definitely excitement there. And these are microorganisms that have to coexist in a community, and so one can imagine that they might have strong selective pressures to try to influence each other's fitness using small chemicals. Yeah. No, it's actually a lethal mutation. But we don't know that so they have other defects as well. So we don't know for sure exactly why they can't survive, but so I should say there's some variation within the population. Some of the mutant individuals can do a little better at clearing the food than others. The time period we're looking at is in early larval life, so they for part of that time they have an endogenous egg yolk as a source of nutrients. So they're able to subsist on that as a source of nutrients for the duration of the experiments that we're looking at. That's a good question.
For us older people, would it help if we spent more time with our grandchildren to pick up young microbes?
Probably. Well, it might. And maybe it's useful for you to be transferring your microbes to them, to the new generation. Yeah. All right. Well, thank you for your attention.